Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another experiment in a preservative series, that is your detection of the benzoic and benzoate, and along with it we are also detect the salicylic acid. Here the preservative are what? Preservative are simply a chemical which is used to extend the shelf life of the food product. Okay, but if the preservative are added beyond the limit of the which are permitted by the FSSAI, then it will come under the category of the adulteration. Okay, that's why this preservative are added in a very appropriate amount. Okay, we all know that this benzoic acid or benzoate are used to inhibit the growth of the different microorganisms, that is your bacteria, yeast and mold, and increase the shelf life of the product. But due to its negative health impact on the human body, the use of this benzoic acid or a benzoate in a many reasons are inhibited, are not permitted. Okay, the government are the food body are not allowed to use of this benzoate or benzoic acid as a preservative. Okay, so this benzoate or benzoic acid also have what? Have a negative health impact. So for performing this experiment, we required a two chemical. One is your dilute HCl, that is 1:3, which means that if we take a 1 ml of that HCl, then we take a 3 ml of the distilled water and we convert that concentrated HCl into the dilute form. Now, when we are handling that concentrated HCl, we have to take extra care, we have to take an extra precaution. We have to wear a proper gloves and proper mask and proper lab coat, so this concentrated HCl does not come in direct contact with our skin. Otherwise, it will damage our skin, it will absorb by our skin, and it will cause a irritation inside our skin. Okay, and also this concentrated HCl also damage our eyes, so we have to avoid it. The second reagent that we required is a 0.5% of the ferric chloride. So for the preparation of this, we required a 0.5% of the ferric chloride, and it will be dissolved into a 100 ml of the distilled water. Then we required a conical flask, beaker, filter paper, dropper, funnel, micropipette of 1000 microliter or 1 ml, micropipette tip, and measuring cylinder. Now what we have to do, we take our 20 ml of the sample, and in 20 ml of the sample we are going to add our 2 ml of this dilute HCl. Okay, this dilute HCl are added into the milk for the acidification of the milk. Now we continuously mix it until a curdle are formed. After that, we filter it by using a filter paper, and then we collect the filtrate. When we collect the filtrate, after that we add a 2 to 3 drop of this ferric chloride into it. If the benzoic acid are present, then the solution turn into a pink, and if the salicylic acid are present, then the solution turn into a violet. Okay, so this is about the detection of the that benzoic acid and a benzoate and salicylic acid. Now we have to take a sample with the help of this measuring cylinder. We are going to take a 20 ml. Now we have to add a 2 ml of dilute HCl into the milk. This dilute HCl are added for the acidification. Now we have to mix it properly because curd are formed. After that, we have to filter it by using a filter paper. Again, it will come very slowly. See that. We have to collect the filtrate. Now after filtration, we collect that filtrate. Into filtrate, I am going to add this 0.5% of ferric chloride, 2 to 3 drops. If the benzoic acid are present, then this solution turn pink color, and if the salicylic acid are present, then this solution turn a violet color. So see that the solution does not turn any color, it is white in color. That means that our sample does not contain any benzoic acid or benzoate or this salicylic acid. If turned pink, then it indicates that the presence of the benzoic or a benzoate, and if it turn violet, then it indicates that the presence of that salicylic acid. So this is all about the detection of the benzoic acid and benzoate and salicylic acid. I hope you will understand, but if you have any query related to this experiment, you can freely to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms. That is your Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.